As we move into the final speaker in this session, it is my pleasure to introduce Dr. Kitchener-Wilson. Kitch is the CEO and co-founder of Rosebud Biosciences. He is a physician scientist with 16 years of experience merging human iPSCs with genomic and molecular technologies, particularly in the cardiovascular applications. As a Stanford-trained molecular pathologist, Kitch was an attending physician at Stanford for half a decade before founding Rosebud. He holds a BS in mechanical engineering, a PhD in bioengineering from Stanford, and an MD from Duke. It's my pleasure to introduce Kitch and his presentation on scaling stem cell-derived organoids for disease modeling and drug discovery.
Great. Can you all hear me? All right. So I am a co-founder CEO of Rosebud Biosciences. We work with iPS cell-derived organoids. This comes from many, many years that I did in academia. I jumped ship to the detriment of my colleagues there to start a company around this. And I do truly think that this has a place in drug discovery. It's at early days for organoids. We've seen a few people mention them. What we really focus on is scaling them and homogenization, precision, quality control in terms of organoids. And it's organoids broadly. And I'll show you what I mean by that. So I show this slide to all the investors that I pitch to. I think this is really what we do. And if you take anything away from my talk, this is it. We have organoids that are mimicking fetal development. Remember that these are iPS cells. Anything derived from an iPS cell is fetal-like. These are not adult tissues. This is not a 6-year-old person's cells in a dish. However, with genetic diseases, you can frequently find a phenotype in these organoids that mimic what happens either in utero in terms of development or shortly after birth. So we introduce mutations into iPS cells. We have our own secret sauce for deriving them in a dish. I'll show you a little bit about that. And then we do longitudinal monitoring every single day. I would love to do it real-time monitoring someday, but we have longitudinal monitoring every day looking for a disease phenotype. This is what they look like in a dish. So this is a 14-day protocol for deriving cardiac organoids. I'll show you a little bit more of that later. These are just bright field images once a day of a developing cardiac organoid. There's this, as far as organoids, I think that there's a lot of, people say organoids. It can mean a lot of things. It could be cancer organoids, maybe little microbiopsies of tissues. For us, these are actually attached on Matrigel, so they're relatively flat. So they're not necessarily 3D, they're 2.5D, if you will. So they're a structure to them. However, the reason we like these, and this was a 2014 publication that really kind of set the field ablaze, these are attached circular micropatterns of iPS cells, 2 millimeters, 3 millimeters across, all on standard tissue culture plates. They go through embryogenesis. So you can actually see concentric circles. Something laser. No, that's laser. Anyway, that's which one? Oh, that little one. Okay. You can see right here. So you can get endoderm, ectoderm, mesoderm, all in concentric circles here, and these are regular. So that means the thing is not just a homogeneous group of cells, but they're actually going through embryogenesis. So we like this because you can scale these much more easily than the spheroids that everyone thinks about with organoids. The other thing, before I move on to this, the other thing about this, because they're relatively flat, makes imaging much easier. They are static. They're not floating up around. They're not spheroids, so you're not getting necrotic cores with them. Diffusion of nutrients, drugs are through the entire thickness of these 2.5D organoids. They make them a lot easier to work with. So these are some cardiac organoids. I come from a cardiac background, so you will see a little bit more of a cardiac bent to this talk, but we have more tissues that we do. Single cell RNA-seq. You get, because of that, it's going through embryogenesis, all the multiple germ layers. You're getting cardiomyocytes, you're getting fibroblasts, you're getting endothelial cells. There's even some neural progenitors, which are that rudimentary conduction system with them. So they are very diverse cell type, tissue types. So I think of these as tissues, not as homogeneous cell cultures. There's a little bit of a busy slide. The take-home point for this is rodents, 2D cell cultures have been the mainstay for decades, for probably half a century, for drug discovery. You go from 2D to rodents. You do your preliminary tests in 2D, then you go to rodents. What I think is that these organoids are going to bring any complexity, and in the near future, they're going to be that gap between the 2D and rodents. In the future, I'm a maximalist on these things. I think that they are going to replace all of those, and organoids in the 21st century are going to be the primary drug discovery platform. That may not happen in the next 2 years, but I think in the next 10, 15 years, you're going to start to see these being the mainstay for drug discovery. These are rich, heterogeneous, macroscopic as opposed to microscopic tissues in a dish that have mutations of the patient. They show disease beautifully. I think that as long as we can standardize them, agree on particular quality controls as we do with our 2D cultures, that they really are going to be the mainstay. So this is a little cool video. They lit up our laboratory with some fluorescent lighting to make it look like a nightclub. We're very excited about this. This is what we have. We have a robotic arm. We grow our organoids in 96 well plates. And you can see our robotic arm. We have a microscope in the background that's moving them around each day. So there's a liquid handler. They're getting fresh media. They're getting bright field images every single day. Once we put a plate of micro-patterned iPS cells in these plates into this system, it's hands off. Humans are the biggest source of error in terms of culturing these iPS cell-derived organoids. So we've tried our best to remove the human component from actually producing these organoids. So this is automated. It works on weekends, it works on vacations. So it's wonderful. We're a team of 3. That's the entire team there. We can produce these in the thousands. We have a capacity for about 10,000 organoids that we can do simultaneously. And that includes longitudinal imaging every single day, media changes, you name it. So the real idea is to use this as a drug discovery platform. And of course, my co-founder, who is big in AI, we are training AI. What we want to do is train it on bright field images of organoids. This is similar to what Recursion is now doing, and get away from the need for immunostaining, biomarker staining, that sort of thing. So just training on bright fields. That's our long-term aspiration. We're not quite there yet, but I think that's where we're going to go with it. I'm going to give you 3 examples. So this is dilated cardiomyopathy, which is my background from my academic days. Hearts get enlarged. They're not able to pump blood. There's probably 50 genes or more involved in dilated cardiomyopathy. It can also be environmental, all sorts of issues. These folks generally need transplants within a year or 2 of getting diagnosed. Mortality rates are atrocious. About 50% die. There are no targeted therapies for this disease whatsoever. So this goes back into my academic days. I found a gene that's associated with DCM. It was upregulated in DCM, dilated cardiomyopathy. This was now 10 years ago. And this led to the whole saga of trying to figure out what it does. The challenge with this particular gene is called long banker, which is a long non-coding RNA. It's primate specific, so it's not found in mouse. You can't do a mouse model with this. It's also fetal specific. It's only expressed during fetal heart development. So how do we get around actually studying what it did, since it seemed to be associated with DCM? Well, HESC, this is human embryonic stem cells, iPS cells, all the same thing. Derived cardiomyocytes also express that, because remember that they're fetal-like. So I thought, after a lot of work, it looked like migration. It caused cardiomyocytes to migrate based on a lot of RNA-seq data and some other reports that we had seen. So I did a swoon scratch assay, which is usually you do with endothelial cells and fibroblasts. Very unusual to do that with cardiomyocytes because you don't think of them migrating that much. And I found a little bit of a difference. It wasn't very satisfying, however. You can see just a little bit. There's a p-value. I think it was a 0.01. That was probably on a good day. I didn't really think that that was the true phenotype and I needed something better. So what we did is we moved on to organoids. I had a colleague that was working on these micropattern organoids at the time. And this is where you saw this beautiful phenotype in a dish that you could see with your eye. And this is actually the source of the name of the company, because they look like flowers to me. So what's happening here is the green are the cardiomyocytes, all derived from, these, in this case, HESCs. They've been micropatterned in these arrays. This is using an older technology, stencils, but this is something we've come around with later to make it more high throughput. And we're overexpressing this particular gene. It causes those migration of those cardiomyocytes, which you can see kind of like a blossoming flower. And here's our control with the scramble control. This to me is what sold the deal for organoids and their ability to show a phenotype far superior to what you would see with single 2D cell cultures. And this is what tells me that organoids have that macro ability to show disease, which you can almost see with an eye, with a beautiful recapitulation of what a disease of in vivo disease. So we've scaled this up. Let's see if this works. So you see that? So we work in 96 well plates. These are cardioids, differentiating them. Homogeneity is a big deal for us. I'll show you a little bit more about what that means. We've introduced mutations for DCM. So on the left, you can see are our beating disease ones. On the right, I'm sorry, let me see if I can show that again. On our right is the disease lines. On their left lines are wild type. You can see they're kind of squirrely looking. So we're really trying to get high throughput homogeneity between batch to batch with these things. High N is really what we're going for here. This is we'll look at the single cell level, single organoid level. You can get beating rates, you can get magnitude of beating, you can get percent rating, and then you can see the disease at the bottom is the disease isn't beating at all. So beautiful data that you can really get with these organoids. We moved on. You know, starting the company, I did not want it to be just a cardiac company. We're like, there's protocols for liver, there's protocols for kidney, there's protocols for brain as well. This is a brain, I'm sorry, a liver. This is for a disease called Alagille syndrome. You get a paucity of bile ducts in these kids. 60% of these kids ultimately need a liver transplant. There is no targeted therapy for these whatsoever. It's a well-defined genetic disease. However, JAG1 is the actual mutated gene here. So here's a hepatic organoids. I'll just point out, you know, they have vimentin is our fibroblasts. Here is our bile ducts. CD31 is our endothelial cells. Albumin are hepatocytes. So there's a diversity of cell types in these. You can also see secretion of albumin by these. So this is actually the media of these organoids that you can sample and run ELISAs on those. They also metabolize urea, and you can detect urea in the media of these. So they really are functional little units of microtissues. So here is where we introduced a JAG1 mutation that causes Alagille syndrome. And I told you that there's the bile ducts. There's a paucity of bile ducts, meaning these kids aren't able to produce bile ducts and move bile through their livers. And so here is wild type, where you see actually normal ring-like bile duct epithelium. These are a few millimeters across, but you can see that regularly. This is a stain for actual bile ducts. So that's wild type. Down here, you see our mutated line. Virtually gone. There might be a few little hints of bile ducts here, but for the most part, they're gone. This is something you would not see at the single hepatocyte level or the single bile duct level. This is something you see at the macrostructural level. And so this is where organoids really come into play. Third and final example, Fabry disease. So this is a disease, there's actually a lot of interest in this disease in pharma. There's quite a few companies working on this. There's shortened lifespans for these folks, about 50 years for men and 70 years for women untreated. What it is there's it's a lysosomal storage disease, multisystemic. You have a healthy GLA gene. It produces alpha-gal enzyme. It metabolizes this sphingolipid called GB3. When it's mutated as GLA gene, you have low amounts of this alpha-gal gene, and you get buildup of GB3. This could be multisystemic. Typically, what these folks die of is either heart failure or kidney failure, but it's multisystemic, so it can occur in lungs and elsewhere. A lot of interest in this. So we were approached by a pharma company that wanted to model this. Apparently, the mouse models do not recapitulate the disease pathology to their satisfaction. And so we said, okay, we have human organoids. Let's try that. We knew nothing about kidney at the time. We developed a protocol for developing kidney organoids in a dish. We introduced 3 different clinical mutations in the GLA gene that are known to cause Fabry disease. We also have a knockout line. GB3 is our marker for the buildup of that sphingolipid, which causes disease in Fabry disease. And then there's a variety of kidney markers. You can actually get little proximal tubules in here and podocytes too, which is pretty cool. So this is what they look like. On the right is what a healthy kidney organoid with a functioning GLA gene that metabolizes its GB3. This is a stain for GB3. On the left here, see all these red speckles? There's a bit of red background here, but you can see those bright red speckles. That's that buildup of those sphingolipids. So we could get this in this was about a 3 to 4 week protocol from start to finish, and we could produce these in the thousands. So that's been exciting for us. We can now do drug screening and all kinds of things with this. These are human, remember, these are not mouse models. So this is just an overview of what we are focused on as a company. I've shown you heart, I've shown you kidney that we've done. We have this liver program. We have an SBIR funding that, although we'll see if that continues these days. And we do have some interest in Huntington's disease and brain. We do have some preliminary data for brain organoids too, although it's kind of early days for that. Ultimately, I want to do all of these tissues. If there's a protocol for differentiation, you can do modify it for a protocol for organoids. We are not spiking in cells. We're not creating 3 different cell types and mixing them together to create these. These are all assembling by themselves as iPS use in our 96 well plates using our different factors that we add to the media. So relatively simple. If you ask AI, what does this is a Grok actually, what does an automated organoid factory look like? And it gives you this. It looks a little dystopian, I'll be perfectly honest. And I think my father had one of these in the 1970s for playing music. But there's some things about this that I do like that captures where we're going for Rosebud, our company. As a pathologist, you think about precision. And precision is consistent between repeated measurements. And that's always been an issue with biology, and particularly when you're having humans culture these cells. If you remove humans from, I love humans, but if you remove them from the actual work of producing these organoids, you get better batch to batch and repeated measurements. Accuracy, are they close to the true value? There's a very low signal to noise in a lot of biological research. We're trying to increase that delta between signal and noise. And so this automation helps that. High throughput, of course, pharma wants high throughput. They don't want little 6 well plates of single organoids. They want thousands, preferably tens of thousands, to really produce high-end statistical value. So that's what we're going through here. Modular. Technology changes. Microscopes get better, liquid handlers get better, robotic arms get better. I don't want a $4 million box that I have to buy from a company that has everything that breaks down. By the way, I have to send a technician to come fix it, but that is stuck with the same microscope, stuck with the same liquid handler, and I'm never able to upgrade. So I like the modularity that this actually image shows, and that's what we're going for as well as company. Interrogatable, meaning you can change the conditions, change the media, change the factors, change the environment of these organoids to produce different data to create disease organoids. And then real-time monitoring. I really think that it's important to detect disease as these things are developing in a dish. And when is the moment when you can first see that detection of disease and that diverges from health? And a lot of AI is going to be involved in this real-time monitoring. So I think that's where we're going. I'll wrap it up here. I don't think this audience needs any introduction to rare diseases. I tend to use genetic disease for iPS disease modeling. There needs to be a genetic component, in my opinion. If there's no known genetic mutation or gene involved in the disease process, I would be hesitant to get into it if because it could be environmental, and it's tough to decide what is that environmental perturbation going to be to elicit the phenotype that you want. So I call it genetic diseases. And, rare diseases, I think it's about 80% are known to be genetic. So that's my preference, it's called genetic diseases. Our goal, high throughput, precise, accurate phenotyping of the human genome for target drug discovery. And our working hypothesis for this company is that 3D tissue is greater than 2D homogeneous cell types for disease modeling. And human data is better or equal to rodent models. I think ultimately they will be better, and that's our competition, our rodent models, to be quite honest. What these do lack, however, is immune system. There is no immune system in these organoids, and that's true of any 2D culture. So we have to find creative ways to get around that in terms of modeling the immune system. So we're sticking with genetic diseases of actual organs for now. This is my team, myself, Evan, and Aditya. We are a Y Combinator company from 2 years ago. We have a few investors in it. We're based in San Francisco. If any of you are interested in organoids, please reach out. Thank you.